All right, what we're looking at this morning for the first part of this session, and I'm recording it because I didn't do a very good job of it yesterday, which doesn't necessarily bode well for this session, but at least I know where I was making the mistakes, and I'm afraid it was due to lack of preparation. But the way this section appears in the exam is very minimal, and there are very specific questions I'm going to ask, which I'll prepare you for those in the mock anyway, and so we don't need to worry too much. But this is about the control of gene expression. And you've got quite a lot of material in your handout folders on this. The first bit of which, and it doesn't all come in the right order because I put it in there in the order I thought I was going to do it and then decided that wasn't the best order. But the first thing that we're going to look at is the idea of stem cells. Now, stem cells are also sometimes a popular choice of topic for presentation. And I need to check who hasn't given me a choice yet. But if you have a look in your folders, you will find that there is a couple of sides of notes on stem cells, and it just takes you through some of the terminology that's used. And then there is a PowerPoint in there, and I'm going to use that PowerPoint shortly to show you some of the key stuff to do with stem cells. Now, so how do stem cells actually relate to this idea of controlling gene expression then? Well, if you consider where we all start in life, a fertilized egg is a single cell. We call it a zygote. When an egg and a sperm meet, you get fertilized egg, correct term, zygote. A zygote then starts dividing by mitosis. And at that point, and for the first few divisions, you could take any cell in that structure and it would develop into all the cells of the human body, whether that's skin, muscle, brain, kidney, anything. Plus, early on, for the first few divisions, it can also develop the membranes that are associated with a developing fetus. So you've got things like the amniotic membranes, you've got the placenta, and so on. And early on in development, you've got these ultimate stem cells that can produce absolutely every cell in the body plus the embryonic membranes. If you go further down the division route, then you get cells which can produce every cell in the body but lose the ability to produce embryonic membranes. All right? And then the further you go, the more differentiated and specialized, the cells become. So ultimately you end up with cells that can only generate one more time, well, the same type of cell over and over again. And we refer to these cells by describing their potency. Potency is the number of different types of cell they can produce. We start off with cells that are described as totipotent. All right, so totipotent cells are the ones in the first few divisions that can produce absolutely any type of cell plus the embryonic membrane. So totipotent is good, isn't it? All right. So toti means total, potent means potential, if you like. So they've got this total potential. So it's all the body cells plus embryonic membranes. All right, but you obviously don't need to keep producing embryonic membranes all the way down the development route, do you? So how is it then that after a few divisions, the cells stop being able to produce those membranes? Why? Well, they do. All the DNA is still there. You know, if you took one of our, let's say, white blood cells, took the nucleus out, looked at the genes, all those genes are still there, but they're not actually, that's it, they're not switched on. So it's this idea of controlling gene expression that determines the sequence. The next step we get to is, they're called pluripotent. Now, pluripotent stem cells are the ones that we often describe as being embryonic stem cells. And pluripotent ones can just produce all the body cells. And these are potentially really useful. And we can actually make pluripotent stem cells from other cells in the body now. It's possible to wind them back genetically, turn some genes on, turn other genes off, and actually create these pluripotent stem cells, which are essentially embryonic stem cells, the ones that you'd find in the sort of ball of cells that you get early on, and then send them down a different route, perhaps, to create cells that we want. You could, for example, take cells from the skin. Now, cells from the skin you wouldn't think as being stem cells, but we have to keep replacing the skin, don't we? So the lowest layer of the epidermis contains cells that only produce new skin cells. Once we get to that stage, they're called unipotent, but that's a little bit further down the line yet. You can take those, treat them so that they are wound back in time genetically to become pluripotent stem cells, and then treat them differently to become, let's say, heart cells. So it's, you know, it's possible now to switch genes on and off. Umbilical cord blood is, yes, is already embryonic stem cells, and that's why it's a useful thing. You can actually have that. You can opt to have umbilical cord blood frozen, I believe. The problem is that there really isn't very much of it, and it's of limited use, and it costs about 10 quid a year to store or something like that. But yeah, there are donation requests. Yes, I've come across that as well. But this idea of the embryonic stem cells, they've got a lot of potential use, you see, for creating cells that could be used to repair damaged hearts, to grow new kidneys, to repair spinal cord damage is one of the biggies. And that's actually been done using stem cells out of the roof of the nose.
Does that have to be blood type?
The blood type, if you, yes, with any cell, you're still going to have antigens on the surface that identify it, but they would, it depends what the use is being, you know, what the use is that's intended for culturing, or, but O negative is, yes, I mean, because it's a rare type, that means that any transplant involving somebody who's O negative has to be O negative. So that is, and because that's a rare one, it's a useful one to have. So, well, the negative is relatively rare, should I say. Moving on from pluripotent, you then get to multipotent, where you've got cells that are a bit more committed to a particular type of cell. So bone marrow, for example, is multipotent stem cells, because bone marrow produces red cells, about 7 different types of white cells, and platelets. So stem cells in the bone marrow are multipotent. So, yeah. Yeah, I was going to say, yes, that's a good description, actually. Family of cells. Whoops. So, like I say, e.g. bone marrow. And then you end up with these unipotent ones, which are just producing one type of cell. And you think, well, are they unipotent then? Aren't they just that sort of cell? No, because there are some definitions of stem cells that I need you to know. So unipotent. There are other words that are used as well, but we will stick with these for now. Do you hear the word progenitor cells? They, I think, fall into the same sort of category as unipotent. But what we now have is a way of replacing cells or growing new ones, and what we need to work out is, you know, what are the actual characteristics, and how do you know a skin cell from a skin stem cell? All right, we will have a look at this PowerPoint. It's one that I got off a website called Eurostemcell. So, presumably it's down there. Right. I have several versions of this. I've sort of edited this down from its original form. Right. There are a lot of notes associated with this, so we'll put it on. Yeah, I'll put it on Moodle, because if you look down at the bottom here, hang on, just let me move that. Whoops. There are presenters' notes, which I don't use, but each slide has got notes, and so if you want to read more about it, there's information on there. This is also the sort of thing that you can do when you're doing your own presentation, is to put notes on, and you can then print off for yourself slides with note pages, and they don't show when you're doing the presentation, see. Right. Okay, so the idea here then is only an introduction, but from one type of cell, let's assume that's a fertilized egg cell then, a zygote, we can get all these different sorts of cells, and it is only basics. And what happens then? Up in the top left there, you can see success, sperm cell meets egg cell. We now have our fertilized egg. First thing that happens once that's been successful is we get Homer Simpson's eyes. Yeah? Once we've got to the 2-cell stage, we get mitosis of each of those to 4, and then so on, so we get a ball of cells. And then we get the development of the first sort of recognizable embryonic sort of thing called a blastocyst. Yeah?
I just want to double check. So the gametes divide by meiosis.
The gametes don't divide. No, the gametes are haploid cells. The gametes are produced by meiosis that reduces their chromosome number to half. So each of those gametes, the egg cell and the sperm cell, have been produced by meiosis. Each one's got 23 chromosomes in it. Once they join together, you've got your 23 pairs. From there on, yeah, the only thing we've got showing on here is mitosis. And so we're producing identical cells. Now our ultimate stem cell then is the one up at the top, although actually it's not quite, because the sperm and egg nuclei haven't fused. But for the first 2 or 3 divisions, we've got cells that can become whole embryos, whole human beings, plus membranes. And it is possible to actually separate the cells in an early embryo, put them into separate mothers, and get identical offspring. It's a form of cloning. And yes, it's Unit 1 GCSE Biology, so yeah, some revision for Kylie on that one. That yes, it's called embryo transfer, and it is used. It's used in agriculture to produce lots and lots of perhaps sheep or cattle with certain characteristics. You produce an embryo in a dish, you let it divide to 2 to 4 to 8. You then separate those 8 identical cells, implant them into separate surrogate mothers, and you get octuplets, identical octuplets. All right, so if you've got the characteristics you want, that's actually worth doing. Then you separate those 8 out into 8 individuals, put them into 8 surrogate mothers, and you get 8 clones. 4, you can do it. No, you can do it with 4. You can do it with 2, but cost effectiveness, you might as well get 8. I don't know how much further on you could go. I don't know where it stops being possible to produce the membranes as well, but I think 8 is is the standard sort of point, and then after that it's not so clear. But by the time we've got to this level, which is something called a blastocyst, you can see you've got a surface forming. There's an actual sort of structure to this, and inside there we have what we call a cell mass, and that's what will then develop to give us giant baby and quite small adult.
So when it gets to blastocyst,
blastocyst is yes, it's just got more of a structure. It's not as clear as that is going to form the membrane necessarily, because you've got to form all the other stuff as well. I mean, interestingly, outer layers of the embryo actually fold in to give you the nervous system, for example. So, you know, the actual process of embryonic development is really quite complex in itself. So what we've got here then, hang on, I went the wrong way. No, I didn't. If we look at our stem cells, the two characteristics of a stem cell are, first of all, that they can self-renew. That's an important thing, so underline that on your slide. And the other thing is that they produce specialized cells, they differentiate. So those are the two characteristics that you need. If you've got a stem cell, what can it do? It can make these specialized cells, but it can also make a copy of itself. So you've still got one. All right, so those two things, I think they might turn up in an exam question. You know, what are the two characteristics of stem cells? They can self-renew and they can differentiate. All right, they can produce cells that are not like themselves. But they have to produce another copy of themselves, don't they? Right, because if they just produce two muscle cells, for example, then you've got no more stem cells. So they have to self-renew and differentiate. So if you were to look at it in terms of, you know, multiple divisions, then if you've got one stem cell to start with, it does that, produces a specialized cell and another copy of itself, and then does it some more, you've still got one stem cell. It's had to keep producing a copy of itself each time it's also produced a differentiated cell. So those two characteristics are really key to this. It's not quite as simple as that. At the end of this PowerPoint, there's a few slides that show you what we'd call hierarchies, where you go, you know, from, it doesn't go straight from the stem cell to a fully formed, mature nerve cell or muscle cell or something like that. You go through the different stages of potency until you end up with just the one specialized cell. So where do we find them? Well, there are different types, and if we go through the first few stages there, we have what we'd call embryonic stem cells. And these cells are the ones that are most useful for research. They're the ones that have the greatest potential for providing treatment for things like nervous system damage and so on. The embryonic ones, yeah. Embryonic stem cells, yeah, they start off totipotent, end up pluripotent, but embryonic, still useful. And these are the ones that we can make. We can actually make them from mature cells. Well, not, you know, from other stem cells. So skin stem cells, for example, could be turned back to this. Once we get to fully formed individuals, we then have tissue stem cells that are there throughout life. And they're in some curious places, but we'll come back to those in a bit. The embryonic ones, well, we only really find them in the embryo. And you can see on there, it's... Oh, can't get that thing to move off the words. Get it up there. The blastocyst, which is a very, very early embryo, you've got this outer layer of cells, which is just a container, if you like, at this point. But it's the cells inside that are useful. And it is possible to experiment on these. For how long are we allowed to do it? 2 weeks. Yeah, you're right in thinking, you know, it's not for months, perhaps. 2 weeks is just the law that says you can experiment on cells from embryos for up to 2 weeks, then they have to be disposable. Yeah. So where do human embryos come from? Humans? Well, yeah. They are spare IVF embryos that people have donated. But, you know, it is just to the point where they are still a ball of cells. So, you know, it is very, very tightly regulated. But otherwise, you might have people getting, you know, a dozen fertilized eggs out of a treatment. They use perhaps 2 of them. What happens to the rest? You know, you either get rid of them straight away, or, you know, they can be studied for a while and then, yeah. So you can take those cells and then you can actually culture them and look at the process of changing their direction of development. And this is the trick. You know, this is what we want to know more about. How is it that these embryonic stem cells could become all those different types of specialized cell? And it's about turning the genes on and off. Remember, this section is about control of gene expression. We've got pluripotent cells here, but how are we going to get to these different sorts of cell? Well, it's just about growing them in different conditions, and that's what happens in the embryo. You could get, under one set of conditions, you could get skin cells. Another set of conditions will give you nerve cells. Again, a different set of conditions, blood cells, liver cells, who knows?
They're all in the same area.
Yeah.
How do they get
Well, they're within an embryo, you'll get things like concentration gradients that just result from position in the embryo and that sort of thing, and communication between cells that are next to each other. So if one cell starts to develop one way, then the one next to it might develop a different way. And it is just about turning genes on and off. And you know, being able to do this in the lab is actually remarkable. The tissue stem cells, well, where are they? Oh, they're all over the place. Not as obvious as you think, really, but surface of the eye, we have to be constantly replacing that because, you know, every time we blink, we're wearing it away. There are a few stem cells in the brain, although people don't really think of that as a source of stem cells. There are areas where there are, but being able to replace brain cells is not that useful on the whole, because what we lose if we damage our brains is what we've learned. You know, so if you, let's say, suffered a stroke and lost speech, you could replace those brain cells, but you would still have to learn how to speak again. So, you know, that's the problem with the nervous system. It knows its limitations, really. You know, there's not a lot of scope for repair there. Skin has got its own stem cells to produce more skin. And then, other than that, we'd find breast cells. Well, breasts can create more tissue, can't they? Intestines, constantly the inside of that being rubbed away. Next one, I have no idea what they're doing there. Why there are stem cells there, couldn't explain. Muscles do, bone marrow we know about. So there's an odd selection there. I'm not... Well, interestingly, yeah, given that that's an adult outline, the bones in the arms and legs do not contain active bone marrow. So no, the active bone marrow in adults is actually limited to places like the pelvis, the sternum, the ribs, the spine. So yeah, it's an odd place to be pointing. But everybody thinks of bone marrow as, you know, you think of a big femur, don't you? But not in adults. So this is what they can do then. Tissue stem cells can differentiate into particular types of cells, which is what makes them multipotent. So blood ones are particularly good at that, and you can see here we've got a femur shown as the bone in question, which yeah, it just is the most recognizable bone, isn't it? These are the ones that are fascinating, induced pluripotent stem cells, because these are the ones we make in the lab. Well, not we, you know, I've never done it, but
Would you like to?
well, it's, yeah, you've got to have a use for it, really, to be, you know, to want to do it, I think, you know, to be researching medical uses of stem cells. But the idea is you take a body cell and you create it, well, sorry, you turn it into an embryonic stem cell. And we call them induced pluripotent stem cells. Now, it says there, genetic reprogramming, that's the important bit. It's not actually adding certain genes to the cell. You can't do that. That would be genetic modification, wouldn't it? But what you can do is switch on genes that have been switched off. All right, and there's a lot of points at which we can control gene expression. So theoretically, we can make these cells in the lab, and then if we treat them in a particular way, we can make whatever sorts of cells we want. And so I have seen examples on documentaries of creating, say, a little beating heart, a rat-sized heart. I know that there's a bloke, I think in Romania, although I'm not entirely sure now, who has had his spinal cord repaired using stem cells that originated from the roof of his nose, and is learning to walk and all that sort of thing. And there are there are stories out there, but the potential is epic, really. It's amazing. And the beauty of using these sort of cells, of course, you don't need embryos, which is, has got to be a, ethical bonus, doesn't it, really? You can't see those particularly clearly on the big screen, but you should see there is just a difference there. And the idea is you start with, say, a skin cell over here on the left. By genetically reprogramming it, or a group of them, let's say, treating them chemically so that certain genes are turned on and off, you end up with these pluripotent stem cells, which, if you treat them differently again, you can use to create, let's say, cartilage or bone or nerve or skin or muscle or respiratory epithelium. It just could be anything. All right.
So we're going to be using genetic reprogramming
Well,
to emphasize turning genes on or off.
Genetic programming is just a chemical process that's used by stem cells normally, but if we manipulate that, it can be done artificially so that you can create the tissues that you want from, you could say, by the time you get to a pluripotent stem cell, it's like a plain cell that's not anything in particular. Yeah. So this idea of potency is just flagging up there, which we started with anyway. Now, I'm not going to go through this in much detail, but just to sort of give you this idea that, when you turn a stem cell into a specialized cell, it doesn't just go straight from stem cell to mature functioning nerve cell, blood cell, whatever. There are steps in that direction through this level of being multipotent, where you get these cells that are described as committed progenitors. So they have decided to become blood stem cells, that sort of thing. And then there's some examples in here of particular ones, which I'm not taking you through all those, but just, stem cell to actual cell takes a number of stages, neural ones and gut ones, and they're sort of connective tissue type ones. How does it actually happen? Well, it does just depend where the stem cell is. So this sort of answers the question as to, if you've got a whole lot of them, why is it they develop in a certain direction? It depends on what we call their niche. Now, the niche of any living organism, and cells the same, is just what their surroundings are. It's everything that's in their neighborhood. And so the microenvironment around stem cells will determine what happens to them. And there's this communication between the cell and the niche. And you have three options, really, here. The stem cell could be in direct contact with other cells that create a particular direction for that cell. There could be soluble substances that diffuse through an environment that influence the way a cell develops. So you might get one group of cells starting to develop one way. They produce a chemical that diffuses across and causes other cells to develop another way. Very, very complex. And then you've got the potential for intermediate cells there, which receive perhaps information from the environment, and they pass on a message, as it were. So, this idea about stem cells then is all about turning genes on and off. Okay, now there's a lot more to this idea of turning genes on and off, and the next step is to start looking at some of the other processes. And did I put... No, I didn't. I didn't put a heading for stem cells here, did I? What we actually need in here is number 1, stem cells. All right. Now, number 2 is going to be looking at factors that can affect the expression of genes, at that level. So we've now looked at, we obviously see, cells will develop down different routes depending on which genes are turned on and off. If you have a look through your notes, if you go past that PowerPoint, there's 3 sheets on gene expression which cover quite a number of things. Just some background reading. I always tend to put a lot of material in when I'm just starting a new topic. So like with the physiology unit, there was a lot on the kidney that we didn't need it all, and over the years I then sort of refine the handouts. You will see after that you've got a page headed up gene regulation in bacteria. Might or might not do that this morning. It's not something that we need. Then there's the myth of junk DNA. And then there's a page headed up control of gene expression, and that's where we're going next. All right, at the top it says page 62. You also have page 63 following that, which I'm going to use. And then for some reason I didn't include page 64, which we do need, which I've now printed off. So you'll be getting another page to go in there. But yesterday I didn't realise I was missing page 64 until I got to the bottom of page 63. So where we are going with this is, the next step is to look at factors that can actually control the process of transcription. Now, if you remember that back from biochemistry, you're doing well. The transcription factors sometimes transcriptional factors. These are proteins which attach to the DNA to actually enable transcription to start. What you have on a piece of DNA, hang on, before we do that, there's quite a neat little sort of flow diagram sort of effect at the top of that page that takes you through the whole idea of gene expression, where you get DNA transcribed into pre-mRNA, pre-mRNA then spliced to give you the mRNA, mRNA turned into a polypeptide chain, and then the functional protein. So we went through all that before, but all those little boxes underneath, those 5 numbered boxes, are all points where you can control the process. And the first one says transcription factors control which genes are transcribed. You could then, box number 2, different splicing means different messenger RNAs are made. And I showed you that, didn't I, with a word? Was it, I can't remember what it was, repeating or something? Departing, that was it, where you could actually put different combinations of the letters together. So you could get different, so that's a control thing. Number 3, messenger RNA molecules can be destroyed quickly or slowly. And then there's 2 others as well. We're going to look at the first one, and eventually we'll look at the third one as well. But this idea of transcription factors then is what the rest of that side is about, and the top part of page 63. And it focuses particularly on estrogen, and that's going to be our example. There are loads of other examples, and we have examples in bacteria that you've got notes on, which I think we will try and avoid putting too much extra detail in this morning. But the idea of a transcription factor revolves around the idea that, and I'm really just going to elaborate a bit on the diagram that we've got here. When we were talking about protein synthesis in the biochemistry unit, we said, right, here's a gene. It's a section of DNA. In order to create the protein from that section of DNA, you have to separate the 2 strands, and you then get RNA polymerase comes along, it builds messenger RNA on that, against the template strand. The messenger RNA goes off, and that's used to make the protein. Well, it's not as simple as that. How is it that the messenger RNA is produced just from a very specific point? How is it that the RNA polymerase knows where to attach? How is it knows when to start? This is about gene control. The expression of genes has to be controlled, because every cell in our body that's got a nucleus has got a full set of genes for operating a human being. So your brain cells, for example, have got the genes for insulin production. Well, it's no use to them, is it? So they don't work there. So they're not turned on in the brain, but they are in the pancreas. Whereas cells in the pancreas don't switch on their genes for producing adrenaline. So, you know, there is a lot of control going on, and so we don't use all our genes all the time. And even with insulin production anyway, we're not producing full quantities all the time, are we? We produce it in response to blood glucose levels going up. And then when blood glucose levels go down, that makes the insulin gene switch off. So we've got this control process going on, which is it's quite interesting, but phenomenally complicated overall. So what you have in a gene, a simple way of looking at a gene, is that if this is your piece of DNA and your gene is here, that's the bit that you want. There's a lot of other areas of the DNA that appear to be important in all of this. And you'll find that you've got, just before the gene, you have another area called the promoter, which is a sequence where the RNA polymerase can attach itself. So RNA polymerase won't just go and try and find a gene. RNA polymerase attaches to a promoter that is just before the start of the gene. That makes sure that it starts at the beginning, right? Not halfway along. So the RNA polymerase can sit there. But it won't necessarily start copying until you've got transcription factors doing their bit. And if you look at the diagram, you'll see that they've drawn 2 transcription factors on here. Very often there are more than 2, and they form some really, really elaborate shapes. And I'm going to leave it simple this year, but in future I might use some much more complicated diagrams than this, I think. But these are transcription factors. And the way it's described in the diagram that you've got, and I don't want to do it any different because you've got a good set of notes there, but these transcription factors will bind to each other, and they will actually sort of activate the RNA polymerase, if you like. So you get a loop forming, a bit like that, where you've got the transcription factors interacting. And that allows the RNA polymerase to then do the transcription. No. So you can also then have other molecules that will stop that happening. And there are all sorts of on-off switches. There's some really good examples in bacteria, but yeah. Yes, these transcription factors are proteins that bind to the DNA.
And they're there all the time?
They are there in, yes, an unbound form, and it's some of them will be bound. So there will also be some that aren't. You can have, I've seen animations and examples with 7 of these, not just 2. And so you might have some that are bound to the DNA permanently, but they then have to be looped and bound together and stabilized, if you like, by others that come in from outside. And then you might have sort of inhibitors that will also switch it off. So they might come in and undo it. And it's, yeah, this is a very simplistic picture of something that's a, it's an emerging area of knowledge, really. But yeah, it's just this idea of understanding that transcription doesn't just happen, that there are factors that can control this. Now, the example that you've got on the handout there, and the one that, the one that comes off the A-level specification, is to do with estrogen. Estrogen is a transcription factor. Well, it's not directly. I mean, it's a hormone, but it binds with a receptor in the cytoplasm to create a transcription factor. And the unit, or the human physiology unit, because we did the two different types of hormones. We did the peptide hormones, which just attach to the outside of a cell and then throw a biochemical switch, sort of thing. But the steroid hormones, sex hormones are an example, like estrogen, can actually dissolve through the membrane, diffuse through because they're fat soluble. They're then picked up by a receptor inside the cell. Now, when the estrogen binds to the receptor, that's now a transcription factor. So that goes into the nucleus, binds to the DNA, and triggers the process of transcription. It's amazing, isn't it, really? But that's how steroid hormones work, is that they, they get into the nucleus like that, and they act as transcription factors. So, yeah, all of a sudden things get much more complicated, don't they? So the example that you've got, estrogen binds with its receptor in the cytoplasm to form a transcription factor. Oops, a trans factor. I was going trans. I can't get myself to write transcription. Come on, transcription factor. All right, so any of the steroid hormones could do that because they can diffuse through the surface membrane, and that is how they do their job. So you've got a diagram of that as well that shows the estrogen being picked up by a receptor, and you've got a sequence of 5 points there. So estrogen, like all steroid hormones, is a lipid, crosses the cell membrane by diffusion, enters the cytoplasm, binds to its receptor protein, which is called ER alpha. ER, estrogen receptor, using E because that's the American spelling of estrogen is without an O. So ER alpha, estrogen receptor alpha, to form a hormone receptor complex, and that is now a transcription factor. So that gets into the nucleus, binds to a particular base sequence on a DNA promoter, and that sets off the RNA polymerase to transcribe genes as required. So that's another method of control. Now, I've got several others. I'm not going to use the bacterial one for now because I think that's a bit surplus to requirements in terms of what you need for the exam. All right, where had I got? That was just number 2, wasn't it? The third example of control is epigenetic control. So if you've got, let's say you've got some mice, and if they've got a gene for a particular coloured fur, you would expect them to have that colour fur, wouldn't you? Yeah. It's not that simple. You can actually change some of the features of the DNA which will change the colour of the fur, despite the fact they've got the gene for it. Now, this is changes to the DNA that can actually happen in your lifetime. And it's not changes to the base sequence. All right, this is changes outside the genome, hence epi means outside, epigenetic. So this is not to do with the gene, but this is changes to gene expression caused by factors in the environment. And with human beings, it can be things like smoking. It can be dietary changes. And there is a piece of research that sort of backs all this up, based on the fact that in 1944, so we're sort of talking, war years, aren't we? There was a distinct lack of food in the Netherlands, and they talk about the Dutch famine, and there was to the point of people practically reaching starvation. And a study afterwards on the babies of mothers who were pregnant at the time found that mothers who were deprived of food in the first part of the pregnancy gave birth to normal weight, healthy children. But mothers who were deprived of food in the later part of the pregnancy had smaller, lower weight children. Now you might think, well, that's just the nutrients, isn't it? Well, no, because if you look then at the grandchildren, so that's the children of these mothers' children, the same thing happened regardless. So those children would then be perfectly well nourished, including during their pregnancies, but their children were larger or smaller depending on where in pregnancy their grandmothers had had a lack of food. So what that indicates is that the availability of food was actually influencing the DNA.
So wait, they're grandmothers. So my mum's mum,
Yeah.
Big Dutch.
Okay.
She lost 2 children during quarantine.
Oh, right. Yeah.
Twins.
Yeah.
So that would follow on to my mum and then to me.
It could, yes, it could do. The food problem has influenced DNA. It won't necessarily be in everybody, but we're now finding that this idea of epigenetic control, you know, I'll tell you what the 2 main things are that can happen to the DNA. But yes, if you'd got, you know, here's parent 1. Well, let's have a pair, right? You have children from these parents, and then they have children. And what happened to parents at this level is influencing these, all right, in terms of birth weight, regardless of the nutritional status of this lot. Okay.
So your grandparents, it's going to affect more than your parents?
Yes. So your grandparents up here, the asterisk lot, clearly the mother's nutrition during pregnancy is likely to affect her children. We know that. That's just common sense. You know, the food that you eat is what your baby's getting as well. So if you don't have enough food, you quite likely will have a low birth weight baby. But more than that, it seemed to be that it mattered when in the pregnancy the woman was getting a lack of food. As if it was late in the pregnancy, it affected the birth weight more. Or you could say, yeah, right, that's because there's more significant growth at the end of the pregnancy and that sort of thing. But if you now take this generation and they have children, but this generation here are perfectly well fed, they've got, you know, no problems with the nutrition, you still find that if they were low birth weight babies, this generation are low birth weight, which has nothing to do with the nutrition of the parent, but to do with the nutrition of the grandmother during pregnancy. All right, so that's quite a, you know, you've got to sort of link all that together. So this, you know, that's one example, and then this idea of the colour of mice being influenced as well by external factors. I'm trying to think whereabouts this comes. You've got 4 sides, which were supposed to be 2 nice back-to-back handouts on epigenetics. You've got one that's Horizons in Bioscience, Epigenetics, Looking Beyond Our DNA. On the second page of that, you've got a little image of a pair of mice. Yeah? Despite their differing colour and body mass, these 1-year-old female mice have identical DNA sequences. So what you would do there is produce identical twins by, you know, getting an embryo, splitting the cells up, putting them into surrogate mothers, like I was saying before, and then treating those surrogate mothers differently. And what you find is that maternal dietary supplementation with a methyl donor, such as folic acid, that's an interesting one, we know about that, don't we, shifts the coat colour of the offspring from yellow to brown and reduces the incidence of obesity, diabetes and cancer. Right, but they've got the same DNA. They're identical twins, effectively, but put into different surrogate mothers. Those surrogate mothers are treated differently, and they come out that different. It's amazing, isn't it? Now, the important thing there is it talks about maternal dietary supplementation with a methyl donor. Can you remember what methyl is from chemistry?
I know it's blue.
That you're thinking of, yeah, methylene blue. What a methyl group is from the naming bit. How many carbons? How many carbons in meth? One. Yeah, CH3 would be a methyl group. Right. So we've got two things that seem to have an influence. There are others, but there are two major. I don't think the picture would be any use at all. The two major external influences. It means external to the genome. First of all, the DNA itself can acquire methyl groups. Now, methyl groups can be added or methyl groups can be removed. And this idea of DNA methylation can actually change the expression of genes. And that's what you see with those mice, right? That methyl groups are being provided. The methyl groups will attach to the DNA. They will not change the sequence of the DNA, but they change the way that the genes are expressed. I don't know whether they attach to the sugar, be somewhere on the backbone. Yeah, the sugar's probably a prime target for that. The other bit that can be affected, rather curiously, are those proteins that the DNA is wrapped round when it condenses. Those proteins, the histones, that's right. Now, histones tend to get, well, they can be methylated, but the other sort of group that's added on there has two carbons. Now, we'd call that an ethyl group usually, but we don't usually say ethylation or ethylation. They still use the old term for this, which is acetyl. So acetylation or acetylation. So it's just these groups being added to other aspects of the genome. Well, it's outside the genome. Yeah, yeah. So it's adding their C2H5 groups. C25. Yeah, jumping ahead of myself. C2H5 groups. And for the methylation, that would be adding CH3 groups. So, you know, if you've got a piece of DNA, here's our piece of DNA, then, you know, if you just add some of these on, then you can influence the way the gene's expressed. Now, on one of those websites that I gave you, I think it's the Learn Genetics one, it could be the other one, there is a virtual lab thing on epigenetics. All right, so you could have a look at that, and it just, yeah. Epigenetics is a huge developing subject that, yeah, I think I was a bit ambitious to include it, to be honest.
What website?
Probably the Learn Genetics one. Sorry? Yeah. It's yeah, quite significant. Okay, so we've got three different ways of modifying gene expression so far. I think I'm going to take a break at that point and just let your brains sort of cool down a little bit. I've got one more to do, which won't take very long, I don't think. And then well, the Learn, it's well, let me show you. There's a lot available on that site, and I did suggest you looked at all of it, you see. Yeah. Where is it? Did I move it? Oh, it's there, look. The Learn Genetics stuff. There's all this. All right, you've got quite a lot of stuff available on this site. The epigenetics section takes you through quite a lot, and there is I don't know where that is. Lick your rats. Actually, I think we've been in here, but there's the interactive. This is the one, gene control. So you've got, yeah, probably it won't run on here, knowing my luck with things like Flash and oh, it does. Right, there you go. You've got this is it's an interactive lab thing on epigenetics. This is the GFP gene, which shows the green fluorescent protein, and you can change. I can't remember what's happening there. You'd have to read it, but this is the DNA around the histones, and you can see there's different little tags being added on there. I'm not going to do that. You can play with that. All right. But there's loads of stuff on both of those sites that can fill this out now. Okay, let's take a pause at that point. So that's 3 mechanisms so far. One final one, which you've also got some notes on, but they disappear at the end of a page. So if you can go back to those notes that actually come from it's like an A-level textbook that's available online, actually. You're going back to the typed out notes that had that sort of flow chart across the top, where it said control of gene expression, that one. Yeah. You've got the those 5 points where you can control the process. You've got control of transcription by transcription factors. Then, after that bit on estrogen, it says control of messenger RNA by RNA interference. RNA interference is the last little bit that I want to do, but I just need to give you the page 64 that managed to avoid getting copied in all this.
You never replied. Get you to reply.
This is the 4th bit then. RNA interference. It's sometimes just called RNAi. This is just another mechanism that controls the expression of genes, but I include it because it's one that's got potential medical applications. Because if you've got conditions that are down to, you know, a malfunctioning gene, and you can interfere with the way that that gene's expressed, then, you know, you've got a treatment for it. So this involves what are known as small interfering, is it messenger RNAs, or they just call them RNAs? RNAs, isn't it? All right, so it's siRNA. Now, there's a particular mechanism for this, but RNA, of course, is a single-stranded piece of nucleic acid. And these little things, they're small in the sense that they are usually no more than 20 nucleotides long. So it's a piece of RNA, 20 nucleotides long, and it folds into a sort of hairpin shape, and you get a base pairing effect, although it's not a double-stranded molecule. It's a folded, single-stranded molecule. So you get this sort of effect. This is a small interfering RNA. So we'd have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and then a complementary 10 like that. So that's the small interfering RNA. Now, what happens to this is described in your notes there, but it's the first thing that happens is it binds to a particular protein. Proteins again. And I always have to check the name of this. It's called the RNA-induced silencing complex. All right, so this is the siRNA. And this binds to, let's get this right, RNA-induced silencing complex. protein, and it's just known as RISC, RISC. Okay, now it's got a very suspicious, I keep wanting to say, specific. It's got a very specific base sequence. So that specific sequence managed to get itself combined into suspicious there. It's got a specific sequence, and when it binds to this protein, RISC, the RISC actually chops it in half. So the next thing that happens, I don't know quite what order to do this in, but you've got it described in the notes very adequately, and there's a good set of diagrams for this as well. They're the coloured ones at the top of the page I've just given you. So the RISC actually breaks this into the two bits, and you end up with one of the bits stuck to RISC, the other bit just gets discarded, which makes you wonder why it was produced in the first place, but I'm sure that will be answered one day. So RISC cuts, so I'm going to do it in a sort of supplementary sort of set of diagrams and notes. So the RISC cuts the small interfering RNA into two pieces. One stays attached to the RISC, the other one's lost. Okay. Now, what you've ended up with then, the bit that's lost, we can forget all about that, but you're now left otherwise with this RISC protein with a single-stranded bit of RISC. All right, so the RISC has a piece of the small interfering RNA attached to it, and that binds to a piece of messenger RNA which is in need of control. All right, now this is to remove the messenger RNA so that it isn't just used endlessly. So if you think about it, once you've transcribed a gene and you've got the messenger RNA, you know, what happens after that? Let's say you've got 1 million copies of messenger RNA in the cell. Well, you'll just keep churning out that protein. So you don't actually have to remove the RNA so that, you know, there is some control. And this is what that does. It interferes, it silences the RNA by cutting it up. So this, the small interfering RNA allows the RISC to bind to messenger RNA at a particular sequence. And it then cuts the messenger RNA up. So the RISC is the important bit here. This cuts the messenger RNA. So it's no longer functional. And if you have a look at the diagram sequence there, you can see what happens, I hope. So you then get, enzymes will then recycle that piece of messenger RNA. The diagram starts off, number 1 is your small interfering RNA. Number 2, they match up with the numbered sequence in the notes, I should add here, I think. Number 2 is then the binding of that small interfering RNA to the RISC protein. Although it should show it actually being still probably the hairpin shape in number 1. It then gets chopped in two and an unused strand is discarded in that third diagram. But you've still got the big yellow blob, which is the RISC protein, and a single strand of the siRNA attached to it. That single strand of siRNA then allows the whole complex to bind to a piece of messenger RNA, the red thing. It chops it in half, and then you've got bits left, so it stops it from being used over and over and over again. So you see, this is another control of gene expression. It stops you having messenger RNA that you don't need anymore. So you might have, your blood sugar might go up, you churn out all this messenger RNA for making insulin, then your blood sugar goes down. Well, you don't need to make insulin anymore. You don't need that messenger RNA anymore. So you get this happening, it chops it all up, recycles it, and you produce glucagon instead in different cells. Yeah? So it's, it's a good control process. But this can actually be hijacked and used medically, and you'll see some applications of RNA interference down at the bottom there. You can do things like identifying the function of genes in cells by silencing the gene and observing the functional effects. That's, it's quite a good option. Is, if you're trying to find out what a gene does, find out what happens when you switch it off. And that's one way of doing this. You let the gene keep working, but you stop its messenger RNA from actually being functional. So if you give small interfering RNAs to the cell, then it doesn't work. You can genetically modify crop plants by silencing undesirable genes, things like toxins and allergens. And there is an example there to do with the Flavor Saver tomato, but that was not a good outcome. The flavour was not good. It wasn't really, no. You could also, theoretically, treat human patients to silence genes in cancer cells or in antiviral therapies. But yeah, well, because HIV is all about nucleic acids, and HIV contains RNA, so you could chop it up, theoretically, yes. The problem is it mutates all the time, so actually finding a sequence that you could target is not that easy. But there's, the potential is there. Now, the other important side of this, this whole aspect of control, is that, of course, sometimes cells lose control of genes. This is all about switching them on when we need them, switching them off when we don't. And there's a whole area of, what happens when genes get out of control, and particularly those that control cell division, because, of course, they, mutations in those, if they're not controlled properly, will lead to cancer. Yeah. There are two particular types of gene involved in cancer. And they have different roles. They're a bit like in a car, you've got your accelerator and your brake. There are genes in cells that encourage cell division. Now, they're strangely known as proto-oncogenes. Now, the onco bit relates to oncology, cancer. Yeah? Proto-oncogenes can be switched on to cause cell division. So they are cell division genes, if you like. And then there are other genes known as tumor suppressor genes, which are like the brake. They actually help to slow down cell division, so they will be part of the control process. Now, you get mutations occurring in cells all the time, and we're doing a lot of repair all the time. But if you get an accumulation of mutations in both of these types of gene, it would be like driving your car with the accelerator jammed on and your brake cable cut. So, you've got, it's accelerating and you can't apply the brake. So, it's that loss of control. So it's sort of quite you try and jump out possibly, and yes, it's it's about the accumulation of mutations in genes really that causes this loss of control in that sort of case. Okay, let's leave it at that point for the control bit.